Carbohydrates, carbon, hydrogen, oxygen, usually a 1-to-1 ratio. Okay, good thing to remember. It's easy to pick up on those sometimes when you see like glucose, when you see it written out, C6H12O6. If you're not used to seeing that, that can be a good reminder. Oh, that's a carbohydrate. Glucose is our body's, carbohydrates are our body's preferred source of fuel. It's the brain's only source of fuel. Glucose is the only fuel the brain can use. Other areas of the body can use a variety of different molecules, but glucose will be used if it's readily available. Okay. Other than fuel and energy, which is kind of how we think most about carbohydrates, they also have a lot of other functions that we'll talk about as we go. Early on, we're going to see that carbohydrates play a role on the cell membrane, creating some structure there. The primary fuel source molecules, we have 3 monosaccharides, glucose, fructose, and galactose. And then we have our disaccharides, which I'll show you. So this is glucose, our friend glucose. I will never ask you to draw a molecule, okay? The only reason that I put pictures of molecules up sometimes is some of you think this way, right? You know, if you have a chemistry-minded person, thinking about how a molecule looks is the equivalent of telling you what it's like, okay? That's why I put these up there. But it's generally a ring-shaped structure. Rings are very stable, so when you break a ring, just like Lord of the Rings, you get a lot of energy out, right? So we like to use that. Our cells split the carbon into 2, 3 carbon chains, and then those get fed into the mitochondria. The mitochondria create ATP. Okay. In nature, glucose is relatively rare outside of animals. Okay. Plants tend to produce disaccharides, not monosaccharides. Well, a disaccharide is just 2 monosaccharides stuck together. So like sucrose, for example, table sugar, breaks down into fructose and glucose. You get 1 of each. That's a hydrolysis reaction. We'll talk more about the different kinds of disaccharides and this process when we get to the digestive tract in block 2 or 3. All right, polysaccharides. So if a disaccharide is 2 sugars, a polysaccharide is a whole bunch of sugars stuck together, okay? This is called a starch. The body makes these. These are each of these circles in this bottom one is a glucose molecule. If you take glucose and you shove them together and covalently bond them, you get what's called starch, okay? Plants make starch, animals make starch. Animal starches are usually branching, like this one, right? Where you have multiple paths. Plant starches tend to be much simpler and non-branching. Now, why does the body make this? Because if each of these glucose molecules were separate, okay, then each of those glucose molecules is going to create osmotic force because it's a solute in solution. If you get a lot of solute in solution, now you're going to start pulling water towards it, right? Like we talked about, osmosis. So the reason the body creates starches is to keep the molecules out of solution, okay? So by cramming the glucose molecules together, you reduce the amount of osmotic pressure, and therefore it lets us put a lot more glucose in a small space. So this molecule is glycogen, and it's the principal starch that we human beings make. As it's formed, it actually comes out of solution. It precipitates into granules, which means that in one liver cell, we can put a whole bunch of glucose all in one place, right? So these are polysaccharides or starches. This one is just an example of glycogen. Okay, so carbohydrates, that's top. Lipids is next. What's a human being made of? Carbohydrates, proteins, and fats. All right, fats. Lipids also contain carbon, hydrogen, and oxygen, but the ratios are different. Usually it's a C to H ratio of 1 to 2 with very little oxygen present. These lipids can also have a variety of other molecules or compounds present in them as well. Lipids are insoluble in water, so they require special transport systems in our body. You can't just move fat from the digestive tract to the cells because there's a big waterway in between, the blood, right? So we have to have a system for that. Now, in that case, we package this fat into structures called chylomicrons, which kind of hide the lipids from the water and allow them to move around. Much more on that later. So lipids always create problems with our physiology. They're hard to move around. They're insoluble. They also, because they're insoluble, lipids, getting enzymes that are found in water to work on lipid is tricky, right? So sometimes we have to break up pieces of lipid into smaller pieces so that our bodies can handle them appropriately. But lipids contain much more energy gram for gram than carbohydrates do. It's one of the reasons we put up with them, right, is because they're very good at storing energy. Lipids also insulate, pad, and lubricate. Okay, so as you begin your dissections, one of the things you're going to find is anywhere the body needs to pad or fill space, lipid's going to be there, right? Because it's flexible and the body can use it for its energy reserves no matter where it is in the body. If it's packed around an elbow joint or packed around a kidney, doesn't matter. It can still function like lipid while insulating and padding. Lipids are the basis for many of our hormones, okay? The steroid hormones in particular are lipid-based. There's a reason for this. It allows these hormones to pass through cell membranes without needing a transporter, okay? We're going to do membranes later this week, but lipids, the membrane is made of lipid. So anything that's made of lipid can pass very easily through the cell membrane, and the body utilizes that. And then finally, lipids form essential structural components like cell membrane and nuclear membrane, are phospholipids, which without them we would not have life as we know it. All right, so that's a fatty acid, very simple lipid, long chain of carbons and hydrogens with an oxygen and an OH group at the end. We have saturated and unsaturated fatty acids. So this is all single bonds, okay? If we strip away some hydrogen ions and create a double bond, then we have an unsaturated lipid, right? So we have saturated and unsaturated. The unsaturated ones are thought to be healthier. The saturated ones are the ones that are typically found in meat and dairy products. Again, more on that when we get to the digestive tract. Glycerides. Okay, so we don't have a lot of just fatty acids hanging out in our body. Okay, we have some, but this is kind of a transitory state. What we have instead are these guys. We have triglycerides, where we take a glycerol backbone, that's this molecule, okay, and then we add 3 fatty acids to it. We do some fancy organic chemistry, hydrolysis, or a dehydration reaction, right? And then we get what's called a triglyceride. This is by far the most prevalent lipid in the body. Triglycerides are. They're the major component of adipose tissue. And, just pound for pound, we have way more triglyceride than we have anything else. We also have triglyceride that floats around in the blood all the time in chylomicrons. When we get to our clinical medicine track in the spring, you're going to learn all about how triglycerides can lead to things like atherosclerosis and heart disease. All right. Eicosanoids are these fancy molecules. Again, do not learn how to draw an eicosanoid. Okay, not important. But eicosanoids are the backbone for many signaling molecules, particularly in the immune system. So in your immunology adventure with Dr. Grove, you're going to learn about the outcomes of some eicosanoids, leukotrienes and prostaglandins in particular. So they start as this structure. Here's our friend, the steroid. Top one is cholesterol. Okay, cholesterol gets a bad rap. Everybody knows it's associated with heart disease and high blood pressure and such. But cholesterol is also an absolutely necessary compound in our body. We could not live without cholesterol. The issue is we just don't want it to be too high. Okay, cholesterol serves as the backbone for all of what we call the steroid hormones. So if you look at estrogen and testosterone and compare it to cholesterol, you can see some pretty obvious similarities. This ring structure is the same in all three, okay? And the way our metabolism works out, we build cholesterol and then we modify it into estrogen or testosterone, or there's a long list of these steroid hormones that we'll get to in our endocrine chapter at the end. But they all start as cholesterol. Cholesterol also plays a role in membrane health. We're very dependent on our cell membranes to do our fancy human stuff, and cholesterol helps to keep those membranes nice and fluid and easy to move. Then we have our phospholipid. If I had a favorite lipid, it would be this one. Phospholipids are cool because they're kind of like hybrid molecules. They have a head that's hydrophilic and tails that are hydrophobic, which means that if you put them in water, the hydrophilic heads point towards the water and the hydrophobic tails point towards each other. Okay? So you get this structure right here called a micelle, where you have your hydrophilic heads on the outside and your tails on the inside. Now, with a little cellular engineering, you can take this and make it into a cell membrane, because you can put two layers of hydrophilic heads with their tails pointed together and make a membrane out of it. More on that to come tomorrow. Okay? So what is a phospholipid? It's got a phosphate group and a non-lipid group at the top. That gives it its hydrophilic character. Remember, phosphate is phosphorus with oxygen, and that is a pretty polar situation, right? So you got a lot of oxygen molecules right in the middle of this phospholipid, so it makes it very hydrophilic. Then on the tail end, you just have long chains of carbon and hydrogen, so decidedly nonpolar on that end. So the phospholipid becomes the cell membrane, the nuclear membrane, and any of our other membrane structures. All right. So carbohydrates, lipids. Last up is proteins. We spend a lot of time talking about protein, but we're not going to spend a lot of time about how proteins are made. Okay, we're going to cover that mostly in a short review kind of way. I do want you to know it, though. It's one of those facts that basic physiology and cell biology makes up, like, I think it's less than 1% of the pants. Okay, but how proteins are made, in other words, from DNA to RNA, RNA to messenger RNA, to the ribosomes to the protein. Remember that whole spiel from past classes? That's one of those things they'll ask about. So we will cover how proteins are made later on this week. Proteins do all kinds of stuff. They are the machinery of the human being. They provide support and movement. Without protein, our bones would have no shape, and they would have very little strength. They would have very little resistance to breakage. Okay, movement, muscle is protein. Okay, we'll talk about which proteins that's made of, but pound for pound, muscle is almost all protein. Transport, everything that moves in our body that isn't by diffusion or osmosis, a protein was involved. Okay, so that's moving things across the membrane. That's moving things, moving secretions from one compartment to another. All of that's happening there. Buffering. proteins play a major role in buffering our pH. We're going to spend a whole session talking about acid-base balance. We're sensitive to pH changes. We are also acid producers. So if you put those things together in one organism, you can imagine that, okay, there's a challenge, right? We're sensitive to our pH, but we tend to make acid. So it means that we're constantly dealing with our own acid production. Well, buffering is a way for a molecule to capture hydrogen ions temporarily so that pH doesn't go up and down. All right, and proteins play a major role in this. One of the most prevalent proteins in your blood is hemoglobin. Hemoglobin is a very good buffer, so it helps to keep the pH of the blood right at 7.4 unless there's some metabolic or respiratory problem. All right, proteins are our major component that does metabolic regulation. Most of our metabolism is regulated by regulating enzymes, right? Proteins are enzymes. We turn proteins on and off. If we want to make if we want to make a cell do something different, we'll turn one set of enzymes on and another off, right? And that will shift that cell's metabolism. The enzymes are proteins, right? So our proteins have systems by which they're turned on and turned off. Remember the second messenger systems that you've learned about in the past, right? Those are some examples of turning enzymes on and off. We'll talk about a number of those as we go. Coordination and control. Some of our hormones are tiny little proteins, little polypeptides that carry information from one place to another. And then proteins play a role in the immune system. Antibodies, immunoglobulins, are complex proteins that help us fight off disease. You're going to learn more about those than you probably care to in immunology, but you're going to be experts by the end. And then the clotting proteins that allow us to seal up our blood vessels if there's a hole are also protein-based systems, and you'll learn much about those in pathology. All right, so what's a protein? Essentially, a protein is a long chain of amino acids stuck together. Okay, the amino acids this is the basis of an amino acid. You've got an amino group on one side, you've got a carboxylic acid group on another, you've got a hydrogen, a central carbon, and then an R group. The R group is going to be what varies as you look across different amino acids, right? There's lots of different amino acids. There's 20 that occur in significant quantities. They're a bit like bricks. You can put the bricks together in any order that you want. And when the bricks are assembled in specific orders, the bricks interact, right? The Lego bricks talk to each other and form shape and therefore function. So each R group is different. Long gone are the days when I'm going to ask you to describe the R group for a particular amino acid, right? You can go ahead and flush that out. That's the kind of thing that you're going to look up, okay? That doesn't need to be in your head all the time. But I do want you to know what an R group is and how it makes for a protein, okay? So that reaction, that by a peptide bond where we take two amino acids and stick them together, this happens at the ribosome, right? To remember or to help you recall that. So we've got our two amino acids. We're going to do a dehydration. We're going to bond them together, right? Now, they can also be split back apart by hydrolysis, thus the Lego analogy, right? Easy to put together, easy to take apart. And a lot of times, the amino acids aren't destroyed in that process. You can literally take a protein, break it down into its blocks, build it into another protein, all without building any new blocks, right? So it's a very efficient system. Now, in the end, what we get is we'll get a long chain. Those chains, so there's our long chain of Legos. The different bricks interact with each other. Here you see that A2 and A6 are sort of attracting to each other, and A1 and A5. Well, those attractions in space then fold the protein into a shape. All right, so we have our primary structure is the linear chain. Secondary structures are alpha helix or pleated sheet, in other words, very basic organizations. And then from there, proteins continue to fold into their tertiary structure. The tertiary structure is the final protein shape for most independent proteins. And the shape of the protein and the characteristics of each region, okay, give the protein its characteristics, its function. Now, some proteins work together. So, like hemoglobin is actually 4 tertiary structures all crammed together with an addition of 4 heme units into one big globular protein. We call that a quaternary structure, where multiple proteins are sort of bound together into one functioning unit. Okay, hemoglobin is just an example, and you're going to learn more about that than you care to in the respiratory system. Okay, so some of our proteins work as enzymes, like I've been saying. Enzymes, they catalyze reactions. They make reactions happen that would never have happened otherwise, or at the temperature and concentrations that we have in our cells. Both synthesis and breakdown. Okay, so some general characteristics of enzymes that I want you to be aware of. One, they're specific. An individual protein will only catalyze a small number of reactions. This is important because in a cell, you've got a lot going on in a small amount of space, right? So if every enzyme did 100 different things, there would be no control over what was happening in the cell, right? So each enzyme only does one or only a very small number of things. So when that enzyme is present, that thing's going to get done. When that enzyme is not present, that thing is not going to get done, right? Very simple. So they're specific. They are saturable. Proteins are things that exist in the universe. Like there are so many of them. You know, there's 1,000 of them in this area of the cell. Saturability is the idea that because each molecule, each enzyme, each protein can only do so much, that if you have a lot of things to do, at some point, all of the proteins are working as fast as they can. Adding more stuff doesn't make anything happen faster. Okay, that's saturability. Probably the best example of this is glucose transport in the kidney. Okay, so when your blood sugar goes up, you start spilling glucose into your urine. Why? Because in your kidney, you have these little pumps, these little proteins that pull glucose from the urine and put it back into the blood. Well, if you send enough glucose through the kidney, all of those transporters are working as fast as they can, and there's only so many of them. So past 300 milligrams per deciliter, you're going to start seeing glucose in the urine because the transporters can't get it all out. They're all saturated, right? So it's an example of saturability. And then regulation. Our metabolism depends on regulation. Each cell is doing more than one thing at a time, and each cell has to be doing its part for the larger whole, too. So it means that enzymes need to be able to be turned on and off. There are multiple mechanisms for this. The first is whether they exist or not, right? DNA to RNA to protein, that's a controlled system. Your skin cells don't make the same enzymes as your liver cells do, all right? Why? Because the genetic machinery is never activated in the skin cells in the same way that it is in the liver cells. So that's just an example. Enzymes are regulated. They have to be able to be on, off, turned up, turned down. A lot of physiology falls into this category, where we're going to be talking about regulation of body functions, and specifically that's occurring because of regulation of proteins, okay, and their enzyme structure. All right, so I like this picture because it's very simple description of one way an enzyme works, right? So here's our enzyme. Substrates come in, and because of the shape of the enzyme and the chemical properties of the active sites, okay, the substrates bind to the enzyme in a particular way, right? Look how they're nice and lined up, so these two surfaces are right next to each other. If this enzyme were a different shape, this would never happen, right? What if the active site was here and here? Well, those substrates are nowhere near each other, right? So they're not going to be able to react. So protein shape and the chemical features of the active sites are critical. We're putting our substrates very close together. We're binding them to the enzyme. That means these two things can react very easily because they're nice and close. We get our product form. When the product forms, that changes the chemical nature of the binding sites of the product, meaning it gets released. Now this enzyme is unchanged from the beginning, so it can go and do this again, right? So it's just an example of the elegance of this system. By using shape and chemical properties of an area, we can make reactions happen that wouldn't have happened otherwise. This is essentially the essence of biochemistry in a nutshell, right? And this is not a biochemistry class. I'm going to use what you know about biochemistry, but it's not going to feel like a biochem class by the time you get into it. All right, so enzymes are reusable, but they are delicate. In other words, they have a specific environment that they work in. If you change the pH, they'll stop working. If you change the temperature, they'll stop working, right? Sometimes if you change the calcium level, they'll stop working. So they're delicate, but they have to have all their environmental needs met. An enzyme that loses its shape, we call that denaturation, and it doesn't work at that point, right? So lots of ways to denature a protein. Your stomach does it with acid, okay? When you eat, your stomach acid uncoils the proteins that you eat so that they can be broken down. But it also means that they no longer work right. And then, like I said, we're sensitive to lots of things: temperature, pH. Any environmental change is likely to change the working of the protein. All right, reminder about ATP, adenosine. Adenosine is our energy dollar, right? It's what the cells use to do energy requiring work. So, do you need ATP to make diffusion happen? No, you do not. Diffusion is free. But if we're going to move against diffusion, like if we're going to move from an area of low concentration to an area of high concentration, right? We're going to go up our concentration gradient. Guess what? We got to pay for that, right? And we'll use ATP to pay for that. So what has evolved on Earth is this really elegant system of sort of energy economy, where we take a nucleotide, adenosine, and we add phosphates to it, and we create this very high energy but relatively stable at body environment molecule, right? So ATP, 3-phosphates. The most common reaction that's seen in the body that releases energy is this, where we take ATP and we break it into ADP and P, and energy is released. Now, the beauty of this system is it's reversible. If we put energy back in, ADP can be added to P and we get an ATP again. So this is very efficient. We're not recreating the adenosine. We're not even changing the phosphates. All we're doing is changing how they're attached to each other. So this is a very efficient, very robust system for managing energy. Going from ATP to ADP is sort of like the $1 of cellular energy. You can also go to AMP, like in other words, do this again without take the ATP and break it, and then take the ADP and break it. but each time that you have fewer phosphates, you get less energy out. So this is the most common. Now, occasionally a student will ask, well, why not make it AQP, right? Why not just add a fourth one? It becomes unstable at that point. So ATP is both stable and easy to break, so it's what our body uses for that process. All right. So carbohydrates, proteins, fats. We talked a little bit about energy. Last topic for today will be nucleic acids. Okay, so similar to proteins, nucleic acids are long strings of building block molecules, long strings of Legos, right? But here, instead of a whole bunch of amino acids, there's only 4 kinds of Legos. Now, the biggest difference between nucleic acids and enzymes in terms of function, nucleic acids don't really do anything, but they contain and store information, right? So proteins make machines. Nucleic acids make programs for building machines, right? So they make or they keep the architecture, the blueprint. So that's your DNA and your RNA, which I know you've been learning about for quite some time. So you've got your purines and pyrimidines. You have your sort of generic, like I said, don't memorize the molecules. This is just a refresher of how this all works. Okay, so we take 2 nucleic acids and we can combine them together in a dehydration synthesis. We do this lots and lots of times and we get a DNA molecule, right? So now we have a template for the building of a protein, right? And I know you've all heard